Greetings, everyone. Welcome to today's webinar. I'm Kübra, representing IVIM Technology, and I'm delighted to have you with us. In this session, we will introduce IVIM Technology's all-in-one real-time intravital, confocal, and two-photon microscopy system, a cutting-edge platform designed for in vivo cellular-level imaging of internal organs in live animal models of human diseases. One of the system's standout features is its AI-powered AI image denoising capability, which significantly enhances image quality by reducing noise while preserving intricate cellular details. We are also excited to announce that our AI image denoiser software is now available as a standalone product. So you can apply its powerful denoising features to your existing imaging data as well. Now, I'm pleased to introduce our distinguished speakers to today's webinar. First, we have Dr. Yong-Gyu Yoon, Associate Professor of Electrical Engineering at Korea Advanced Institute of Science and Technology, aka KAIST. He leads the Neuroinstrumentation and Computational Analysis Lab at KAIST, an interdisciplinary group advancing neurotechnology, optical imaging, and computational methods. In his talk, Dr. Yoon will introduce support, which is statistically unbiased prediction utilizing spatiotemporal information in imaging data, a self-supervised novel learning technique for denoising microscopy images. This approach enables clearer visualization of dynamic cellular activity, even in noisy or fast-moving imaging scenarios. Although Dr. Yoon could not join us live today due to a family commitment, he has kindly provided a recorded presentation for this session. Thank you for your understanding. Following this, Dr. Pilhan Kim will discuss the system's capabilities in live in capturing real-time cellular dynamics with high precision. Dr. Pilhan Kim is also a professor at KAIST and the founder CEO of IVIM Technology. His main research interest is the development of advanced state-of-art in vivo cellular imaging technology based on a real-time laser scanning intravital, confocal, and two-photon microscope. Finally, IVIM's applications specialist Jenny will demonstrate the real-time performance of IVIM Technology's new AI image denoiser software, showcasing how it processes high-resolution intravital in vivo imaging data effectively. If you have any questions during the seminar, please feel free to use the Q&A box below, and we will make sure to address all of them. Also, as you can see in the chat, I'm gonna share the registration link for the AI image denoiser software demo. And if you're interested, feel free to go ahead and register. Now let's turn to Dr. Yoon's webinar, where he shares his groundbreaking work on the AI image denoiser.
Hello, everyone. It is my great pleasure to be part of this webinar series organized by IVIM Technology. Today, I'm going to share one of our recent work, support, which is a self-supervised learning framework for robust denoising of microscopy images with fast dynamics. So let me start with briefly introducing myself. My name is Yong-Gyu Yoon, and I'm currently an associate professor of electrical engineering in KAIST. And I've been working at the intersection of neuroengineering, optics, and machine learning. And what I'm going to present today, support, is basically a machine learning work. Before I talk about the details about support, let me start with a little bit of background, which is about the voltage imaging. Okay, so voltage imaging is a class of functional imaging technique, which in general allows us to see the activity of neurons at the single cell resolution. It is very crucial for understanding the interaction between the neurons, and it's also a key technology for system neuroscience. So let me start with a little bit more popular or conventional form of functional imaging, which is calcium imaging, which uses genetically encoded calcium indicator or GESI. So if you do calcium imaging, you get something like this. You see the neurons that are blinking, which corresponds to the neuronal activity. So neuronal activity is associated with the calcium concentration change, which is visible due to this genetically encoded calcium indicator for the following reason. So basically, the genetically encoded calcium indicators, those are the fluorescent proteins that can be bound to calcium ions. So when it's bound to calcium ion, its morphology changes and it becomes brighter. So what that means is when the intracellular calcium indication or calcium concentration increases, the average brightness of these GESI molecules are going to increase. So the brightness basically indicates the calcium concentration within each cell. Hence, you can see the activity of each neuron. So that is about the calcium imaging. And voltage imaging is kind of similar but slightly different. So we use genetically encoded voltage indicator, GEVI, for voltage imaging, which is kind of similar but slightly different. So GEVI is also a kind of fluorescent protein, but unlike the GESI, its morphology changes in response to the voltage across the protein. So what that means is their brightness is going to change as a function of the voltage instead of changing as a function of the calcium concentration. This basically allows us to directly measure the voltage signal within each neuron. So here we have an illustrative example. So as the voltage within each neuron changes, the fluorescent intensity is also going to change accordingly. Okay, so that's the voltage imaging and the genetically encoded voltage indicator used for voltage imaging. And here we have an actual example of voltage imaging. So this is kind of what you can get with the voltage imaging. So this is the voltage imaging data from larva zebrafish. You're seeing the spinal cord of a fish, and this was acquired with light sheet microscopy, imaged at 400 hertz. So in fact, this is actually one of the best voltage imaging data in terms of data quality I've ever seen. But still, you cannot really see the neurons blinking, mainly because of the low SNR of this video. So it'll be really nice if you can improve the SNR of such video through some algorithm or some computational approaches. And support, which is what we developed, is something for exactly that purpose. So in a nutshell, it's basically just a machine learning algorithm for denoising voltage imaging data. But as some of you might be aware of, using machine learning for denoising is not really a new thing. It has been under the research for more than 7 years now, and there really has been a lot of successful works. So for example, in 2018, Noise to Noise was published in ICML, and 2019, Noise to Void was published in CVPR, and these papers really built the foundation of machine learning for denoising. But in a nutshell, these algorithms don't really work for voltage imaging data. We'll talk about the reason later, but it doesn't really work. And there also has been a lot of works, a lot of machine learning algorithms developed specifically for fluorescent microscopy images, including DeepCAD, published in Nature Methods in 2021, and also deep interpolation, also published in Nature Methods in 2021. And the following works like a DeepCAD RT, published in Nature Biotechnology, and the Deep Semi, which was also published in the Nature Methods. But again, while these were developed specifically for fluorescent imaging data, they don't really work on voltage imaging data. So we'll talk about the reason and also how we can work around that. So to do so, we're gonna first discuss the overarching philosophy of all of these machine learning based denoising algorithms. So here we have a face of a human, and we can see a red dot here. This part is hidden, but you can actually imagine what we were expecting behind this red spot. it's going to be white pixel basically, right? And we know that because we can recognize that this is a human face, and we can recognize that this is the eye part, and we know that this part is supposed to be white. So hence, we know that this pixel is going to be white. So let's try to say that in a little bit more computational word. So let's first pixelize everything. And then what we are looking for is something like this. So the hidden pixel, let's denote that as x, and let's denote the surrounding pixels around x as omega sub x. So then what we're seeing is we can predict the pixel value of x because we understand the relationship between omega x and x. We understand the omega x part tells us that x part has to be the white part of the eyes, and hence we know that this pixel has to be white. So we're basically looking for some mapping between this omega x and x. If we're given omega x, we're basically looking for the most likely value of pixel x, considering all of the given information. So in terms of machine learning, what we are trying to do is we are trying to train a neural network that takes this omega x, the adjacent pixel values, as the input, and gives us the clean pixel value x hat. And if we actually have the values of this x, which would be the case because we have the image, then we can basically try to minimize the difference between the predicted output and the actual pixel value at the center, which is x. And we can have a lot of these pairs, omega x and x. We can just do that by sampling different region of the image. So we can have a lot of input-output pairs, and we can just train the neural network so that when it takes the omega x, it's going to give us x hat, which is very close to x. So this way, we can train the network to learn the relationship between the adjacent area and the center pixel value. Okay, so that was for clean images. We're looking for denoising images, right? But the story remains the same for noisy images. So here we have a noisy image. We still have a hidden pixel here. And let's pixel thing. And now we're trying to predict the center pixel given the surrounding area, which is all, again, omega. In this case, let's denote the center pixel as y because it's noisy. And let's denote the surrounding area as omega sub y. So we're trying to predict the clean value, which would be x, given omega y instead of omega x. Again, we're looking for something like this. We are looking for a neural network that takes the noisy neighboring pixels, denoted as omega sub y, and we want this network to predict x value, so x hat, which is very close to x. But we cannot train the network this way because we do not have access to the clean value x. We only have the noisy pixels y because that's a part of the image. But the good thing is that we can train the network using only those y pixel values. So we can basically train the network to predict the center pixel value y sub hat that is close to y. And the short conclusion is that it's going to give us the denoised pixel value x, which is very close to x. And here's the reason. So let's say you train a neural network that can predict the value of the dice you're throwing. So, I mean, if you throw a dice, the value is going to be between 1 and 6, and it's going to be purely random. So there's no way the network can really predict the actual random value, right? So the best the network can do is just to predict the expected value, in this case, which would be 3.5, because that's the best guess that the network can make, again, because it cannot predict the actual values, which is purely random. And in this case, if the network does the same for noisy images, it's going to predict the expected value of the image. And what that means is it's going to give us a noise-free value because these Gaussian noise or Poisson noise, their expected values are 0, meaning that if the network tried to predict the expected value, that's going to be very, very close to the clean value, x. So that's the underlying philosophy of most of these denoising algorithms. And now we're going to talk about why they fail at denoising voltage imaging data and how we can solve it. So here we have an illustrative representation of denoising the time-lapse image data. So here we have a representation of the raw data. So this axis right here, that corresponds to time axis, this axis that corresponds to the space axis, and this axis corresponds to intensity. So these are the pixels, and along this axis, we are seeing the change of those pixel values along time. So whenever you have something like a calcium imaging data, you have something like this. Along the pixel value, the value rises and decays, but it's very slow. The kinetics is very slow, and this is what you're getting. And if you were to denoise this kind of data using like noise to noise or noise to void, which were published in the machine learning conferences, what you're essentially doing is you just take a slice of this, which is one frame, which is a single image out of the video, and you hide one pixel like this. And what you try to do is you train the network so that it can predict this hidden value given these values. So what it's essentially doing is it's trying to predict the whole frame given the surrounding frames. So for the same reason, this predicted frame is going to be denoised. It can only predict the expected value. And as the dynamics is kind of slow, the adjacent frames are very similar. As long as you image at a very high speed, you can actually predict the signal at these intermediate planes. So that's why you can get the denoised result. But it doesn't really work for the voltage imaging data. So when you do voltage imaging, first of all, it's going to be extremely noisy because you have to image things at something like at least 300 hertz or up to 1 or 2 kilohertz. So it's going to be very, very noisy. So if you try to take just one frame and hide one pixel and try to predict it given the surrounding pixels, you have very limited information. So it becomes extremely difficult to predict it. So this was for these papers published in machine learning conferences. And if you were to use these other methods that are published in Nature Methods or Nature Biotechnology, again, you're trying to predict this frame given the surrounding frames. Now, the problem is that the event was extremely short because the kinetics is fast. And what that means is it's extremely difficult to predict what happened using the other frames because the event was too short. So you don't have the information to predict it. So the obvious solution to this problem would be just in that way, you can provide enough information to the network so that it can predict the accurate value so that you can get the accurate denoising outcome. So visually, you're doing something like this. You feed the center frame with the blind spot and also feed the adjacent frames. All of these are the input to the network, and then the network will try to predict the center pixel. But for the reason I explained using the dice examples, it's going to give you the clean value. So since you do this for fluorescent images, this is what you're actually doing. You feed this time-lapse image into network and you give the center frame with the surrounding region and also the other frames, and then ask the network to predict this fixed pixel. That's going to give you what you wanted. To do so, you're going to have to design a neural network that cannot see the center pixel. So for that, you design the neural network with this specific architecture, but I'll just skip that because that's not really important. And then, so that's basically how we developed support, and we applied it to voltage imaging data, and this is what we got. So unlike the previous example, so here we have the raw data, and here you have the denoised outcome. You can actually see the neuronal activity at each single cell, and you can also see those action potentials occurring at each time point. So everything is now crisp and clean. And you can also zoom into each neuron and see what happens. So here we have 4 regions of interest. And if you zoom into these ROIs, this is what you see. You can clearly see the neurons blinking here. And you can also see the temporal signals extracted from these ROIs. Okay, let me move on to the next example. So here we applied it to voltage imaging data, which is extremely noisy and the dynamics is extremely fast. But that does not mean that we can only apply it to fast kinetics data. You can also apply it to slow kinetics data, and it's going to still work. So here we have the calcium imaging example. So this is the zebrafish brain, and we imaged it using a confocal microscope. And this is noisy data, and after denoising, this is what you get. You can clearly see the cellular structures, and upper side is denoised, and the lower side is the raw data, and everything denoised. And you can zoom into certain areas and see what happens before and after denoising. And again, we didn't assume anything about the data other than the center pixel is related to the temporal and spatially surrounding region. So we can apply it to any kinds of time-lapse images. So for example, we can apply it to a time-lapse image of a moving C. elegans, which is a tiny worm. And you can get a nice clean result. This specific video was acquired using the microscope from IVIM technology. This is the in vivo imaging of a mouse ear skin, and this is another in vivo imaging from ear skin, and you can clearly see that it becomes much better. And this is in vivo imaging of a mouse kidney. And this is also another video from the kidney. And one from the thigh muscle. And if you think about it, we are giving x, y, t axis tensor as the input to the network. And it doesn't really have to be x, y, t. It could be x, y, z, meaning that we could just fit in 3-dimensional data to the network for denoising, which is what we did here. This is the expansion microscopy image from Professor Jaebom Jang in KAIST. We applied it to denoise the Z-stack image, and it just magically worked out. And again, you can just do it for any kinds of image. Here we have the two-photon microscopy image of a mouse brain. It's also Z-stack, and we are moving along the Z-axis, and we can see that things are nicely denoised. And in fact, it doesn't even have to be optical microscopy images. We can apply it to electron microscopy image, and we can still nicely get these results. So on the left, you have the raw data, and on the right, you have the denoised outcome, and you can clearly see that the outcome is much better after denoising with support. Okay, so that's basically it for the seminar. I would like to acknowledge everyone who contributed this work, including our lab members and the collaborators. Thank you, and hope you enjoyed this webinar. Bye-bye.
We thank Dr. Yoon for his insightful presentation. Now we will shift our focus to the practical applications of this AI-powered image denoising technology in intravital microscopy. For that, Dr. Pilhan Kim, if you are ready, please go ahead and begin your presentation.
Okay, thank you for the introductions. I hope you can see my screen, right? Okay, so today I will explain about the real-time intravital microscopy, and then what's the application of this microscopy, and then how we use this AI denoising software to improve the quality of our intravital imagings. Okay, so to begin with, intravital microscopy is a very unique technique that can provide this very high resolution, microscopic scale resolution live video from the live animal. So compared to other imaging modalities such as CT, MRI, or whole field optical imaging technologies such as IVIS, by using intravital microscopy, we can easily obtain this single cell level live video from the live mouse. So this particular example is 10-minute timeless video obtained from the lung of a live anesthetized mouse model. And then as you can see in this video, we can clearly see the movement of the individual neutrophil inside the vessel, which eventually block the blood vessel here and disturb the blood flow. Well, in addition to that, in addition to the fluorescently labeled cell, we can also directly image the fluorescently labeled drugs, such as fluorescently labeled nanoparticle inside the vessel. So this is one example. So this is a real-time video obtained from the liver of anesthetized mouse after the IV, the intravenous injection of fluorescently labeled nanoparticle through the tail vein. So as you can see, we can beautifully see the real-time delivery of this nanoparticle inside the liver sinusoid vessel, as you can see in this video. And actually, this video is AI denoised real-time video. So as Professor Yoon already explained, his method, the support method, can be a very optimal tool to denoise the very fast kinetic data, such as this real-time video obtained from the live animal model. And then for this one, I will explain a little bit more later. Okay, so to summarize, intravital microscopy is a very useful tool as it can acquire this live cellular-level video in a live animal in vivo with some molecular informations. So to briefly show you, we have this single box packaged all-in-one dual mode intravital confocal and two-photon microscope. And then by using this microscope, we can easily image almost every organ and tissue in a live animal model, and it's in the mouse model. So as you can see in this slide, and all these images were obtained from the live anesthetized mouse model. And then we have been able to image bone marrow, brain. This is a brain tumor, retina, this is skin, and this is the hair follicle in the skin. And then you can distinguish the individual, follicular cell in the hair follicle. And then also various tumor model. So this particular one is the colorectal tumor model. And then we also have been able to image the pancreas, spleen, GI tract, adipose tissue, prostate, and the placenta in the pregnant mouse model, and the lymph nodes all over the body, memory tissue, cell transplant, kidney, muscle, liver, heart, lung, and so on. And then the great feature of this microscope is this is a very high-speed system capable of real-time imaging with a speed of 50 to 100 frames per second with a frame size of 512 by 512 pixels. And then based on this real-time imaging capability, we also implemented the automatic live tissue motion compression function with this microscope. Okay, so let me show you some real-time imaging data obtained from the lung. So with the speed of 30 to 50 frames per second, we can directly image the flowing red blood cell inside the vessel from the lung surface, as you can see in these videos. So these videos, what you are looking at here is vessel in green color, and the RBC, the flowing red blood cell inside the capillaries. So you can see how this red blood cell flows inside the capillary. And then to obtain this video, we stabilize the lung surface by using this negative air pressurized imaging chamber for the lung. Okay, and then also based on our video rate real-time imaging capability, we implemented the automatic motion compensation functions. So this is one example. This is a very typical, tissue motion we can see in the internal organ of a live mouse model. So this motion is basically due to the respiratory actions, breathing airs from the lung, and also heart beating. But by taking this real-time video, we can really see their tissue motion by looking at their individual cell movement by frame. So with the frame-by-frame image registration, we can compensate the tissue motion with very high precision, rather than better than the sub-micrometer precisions. So this is the result. So by doing the frame-by-frame motion compensation, we can acquire this very high resolution video from the moving tissue in an internal organ of a live mouse model. So as you can see, this motion compensation function is very critical to obtain this high quality, cellular-level images from the live tissue in a live mouse model. And in our software, this motion compensation function is built in to be performed automatically. So what you need to do is just turn on the motion compensation function and press the button. And just within a couple of seconds, it automatically generated this motion compensated, high quality images from the moving tissue. Okay, and then also, as Professor Young-Ju Yoon already showed you, this real-time, very fast kinetic real-time video is a good match for the this support method, Professor Yoon introduced, because it doesn't need any pre-training, and then this is an unsupervised model, so thereby we can perform it automatically. So this is the video. So on the right-hand side, this is a raw video. And on the left-hand side is denoised AI process video. So you can directly compare how much SNR we can easily improve automatically by using this AI denoising network. This is another example. So this kind of very noisy real-time video can be easily processed to obtain this denoised video here. Okay, so we can use this denoising software for all these different internal organs. And then based on our motion compensation functions, we can obtain this high quality, timeless video from the lung, as I showed you before. So as you can see, these individual neutrophils make a cell-to-cell contact and make a small cluster and block the blood flow in just 10 minutes. So to give you an idea how we do it, this is the process. So we first intubate our mouse model, and they connected it to the mechanical ventilator to keep the mouse breathing air, and then expose the lung, and then stabilize the lung surface by using our imaging chamber, lung imaging chamber. And then we put it into our microscope, and then find the focus using this animal stages, polarized the animal stages, and obtain the images. And then using the same principle, we can even visualize the surface of the heart surface, the beating heart in the live mouse model in the beating heart. So this is the example. So as you can see, we can directly see the heart beating and the fast flowing red blood cell and other immune cells in the coronary vessel in the live mouse model. Okay, and then let me show you another example. So this is how we prepare our mouse model for the liver imaging. So first, this is the real-time video to see the nanoparticle delivery to the liver sinusoid vessel. So this is the raw real-time video. Still, this is very nice because we can clearly see how this lead fluorescent nanoparticle appears inside the liver sinusoid in real time. But when we do the AI denoising, we can have a much better quality videos, as you can see in here, right? So we can even distinguish individual single nanoparticle inside the vessel. So let me zoom in here. So as you can see, I hope you can see this video as well. So we can nicely see the individual nanoparticle flowing inside the vessel by using this AI denoisings. And another example, so this is also real-time imaging of the photothrombosis in a live mouse model. So in green color, what you're looking at is the platelet in vivo labeled by CD41. So you can see that this real-time formation of the platelet. And then this is also AI denoised video using support method. And then as you can see, we can obtain very high quality, high SNR real-time video based on our real-time intravital microscope and AI denoising software. And there are more examples for long-term longitudinal in vivo imaging by using these three kinds of imaging window method. So first one is the dorsal skin pole chamber, which can be implanted on the dorsal skin of a live mouse model. And then after these implantations, this mouse can survive more than 3 to 4 weeks. And then we can repeat the intravital imaging in the same mouse model. And then also, as I told you, based on our real-time imaging capability, we can directly see this flowing red blood cell inside the tumor vessel using these dorsal skin pole chambers. And then also by using this dorsal skin pole chamber, we can monitor the tumor nanoparticle delivery to the breast cancer mouse model for 1 day, for 24 hours. So this is a 3 time point after the injection of IV injection of this nanoparticle. Green color is the tumor cell. So let me remove the green color. So as you can see in here, so in 6 hours, most of the nanoparticle stays inside the vessel. But at 24 hours, this nanoparticle, get out from the vessel and then nearly delivered to the tumor cell, as you can see in here, and also in here, and also in here as well. So by using intravital microscope and dorsal skin pole chamber, we can repeat the imaging in the same mouse model and then track the drug delivery to the single target cell in the live mouse model. But these are so not just a nanoparticle, we can image all different kinds of drug modalities, such as exosome and antibody drugs. So this is one example. So in here on left-hand side, you can see the delivery of the exosome to the target cell in the liver at 10 minutes and 30 minutes after the IV injections. And on the right-hand side, we repeat the imaging for 4 times and then see the antibody drug delivery to the brain for 48 hours. So on upper panel is the control drugs, so it doesn't really deliver to the brain. But on the bottom panel, this is engineered antibody drugs, so which can be more efficiently delivered to the brain tissue at 24 hours, as you can see in here. So this is another very good example, how we can utilize the intravital microscope to track, to visualize the delivery of the drugs in the live mouse model. And then this is the repeated imaging at day 7, day 10, and day 13, showing the individual tumor cell in green color and the change of the tumor vessel over time. So this is the same vessel at day 7, day 10, and day 13. And then you can clearly see the dilation of this tumor vessel over time with the infiltration of this GFP green tumor cell. And then also we can see we can monitor the treatment efficacies, as you can see in here. So with the treatment, with some anti-angiogenic treatment, we can suppress the tumor vessel remodeling. So as you can see in here in the bottom panel. So without treatment, we can see this huge tumor vessel remodeling over 6 days from day 7 to day 13. And then, so this is the cell transplantation model. So bone marrow cell transplantation model. And then again, we can repeat this imaging at different time point from day 2 to day 10, and then see how this transplanted cell engrafted inside the bone marrow at day 3, and then proliferate from day 4, 5, 6, and 7. And then at this time point, it mobilized to the periphery, so it disappeared at day 8. But at the same time, this neighboring area now newly engrafted with the transplanted cell from day 9 to day 10. So this is also a very good example. We can track the transplanted cell plate, live tissue, live mouse model for more than several weeks. And also this is the abdominal imaging window for the long-term imaging of liver, kidney, and pancreas, and so on. So this is one example. So here we can see the pancreas and spleen. So after implanting this abdominal pancreas imaging window, so what we can do is we can make the orthotopic pancreatic tumor model by injecting this PANCO2 GFP cell into the pancreas, and then repeat the imaging for several weeks, as you can see here, and then see how this tumor cell grows inside the pancreas, and then make the tumor vessel changes over time. So this is the same site, and the repeated imaging from day 9 to day 14. And then you can see how this pancreatic cancer cell grows, and then how the tumor vessel dynamically changes from this time period, from day 9 to day 14. So you can see the very chaotic embryonic tumor vessel remodeling. Then in some area, we can also see the vascular layer pactions due to the tumor vessel, due to the tumor cells growth. Okay, so finally, this is the cranial imaging window method. So in this surgery, what we do is we remove the small portion of the skull and they put the transplant glass over the brain, and they seal the exposed area with the dental cement. So after this surgical preparation, mouse can survive more than several months. So we can repeat the imaging in the same brain area in a single mouse model for more than several months. So this is just one example of the data set we can obtain from the brain. So through this cranial imaging window, we can directly see the meninges and cortex, and then see the vessel, pericytes, and the arterioles. So this is the one example. And then again, based on our real-time imaging capability, we can directly see the IVIS perfusion inside the vessel on the surface of the brain. And then by looking at the IVIS flows, we can identify the penetrating arteriole in the brain surface. And then we can induce photothrombus in the penetrating arteriole. So make the small microinfarctions in the brain. So this is the setup, which we call cerebral microinfarctions. And then after inducing these cerebral microinfarctions, we can continue. We can repeat the imaging for more than a month, and then see how the cells changes and tissue changes during this time period. So we can see the astrocyte, pericyte, and endocellular cells. And then if you look at carefully, so tissue expand, which means edema, and then after that, day 5, it recovered to the baselines, and then followed by the tissue shrinkages, suggesting localized cortical atrophies. So this one. So edema and then atrophy for during the period of 1 month. And then when we look at the edema period at day 3, this is day 0, day 3, and day 5. And then again, this is same vessel, so same locations. So when you look at day 3, we can see the leakage of this 3,000 Dalton dextrans from the vessel. But interestingly, it spontaneously recovered at day 5. So it doesn't leak out anymore. So we performed the intravital imaging for this time period from day 3 to day 5. And then this is the result. So this is a 24-hour, so 1 day continuous imaging of a parasite in this mouse model. And then in red color, we can clearly see this parasite extend their processes along the endocellular cell. And then this is the key cellular mechanism, underlying mechanism for the recovery of the BBB function in terms of vascular leakage in this mouse model. So, and this in the mouse model, by using our real-time microscope, we can obtain very high quality cellular-level videos and images from all these different organs and tissues. Okay, and then this single box intravital microscope is capable of four-color two-photon imaging and also four-color confocal imaging as well. So let me show you several examples. So this is the in vivo result. Another in vivo imaging result obtained from the skin of a live mouse model. So we can clearly distinguish the individual cells, vessels, and the collagens in white color, and the blood vessel in blue colors. And then this is a muscle. So this is a single field of view, 3D images, G-stag images. And then also our microscope is capable of automatic mosaic tile scan imaging and automatic stitching to generate these large area mosaic images from any tissues. So this is another example from the pancreas. And this is a large area stitched images obtained from the pancreas, again, from the live mouse model. And this is the kidney. And then these large area images. And then together with our intravital microscope, we also provide all these various imaging windows and chambers for longitudinal repeated in vivo imaging of various tissues, such as various cancer model, heart, lung, brain, abdominal organ, including liver, kidney, spleen, mammary tissue, pancreas, and uterus as well. So by using these imaging windows, you can easily perform the longitudinal intravital in vivo imagings. So to summarize, intravital microscopy is capable of 3D cellular-level imaging from the various internal organs in a live mouse model. And by using it, we can do dynamic 3D imaging of various cellular-level dynamics in all these different organs in a live mouse model. And also for the drug development, intravital microscopy is a very useful tool as it can enable a direct in vivo drug imaging analysis to analyze the drug delivery to the target cell or drug efficacy in the target cells. So I hope you enjoyed my talk, and if you're interested, please visit our webpage, www.ivimtech.com. Okay, so thank you very much.
Thank you for the presentation, Dr. Kim. And as a reminder, to book a virtual demo for the AI image denoiser software we just released, simply use the link that I shared in the chat box or fill out the short survey that will appear after the seminar ends. Now, IVIM's application specialist, Jenny, will briefly show the AI image denoiser software in action and how to tailor it to your specific imaging data. After the demonstration, we will move on to the Q&A session. And Jenny, I think you're ready. If you're ready, please go ahead.
Thank you, Kubra, for the introduction. Hello, everybody. I'm Jenny Ohm, Technical Application Specialist here at IVIM Technology. I will now be briefly giving a virtual demonstration of IVIM Technology's AI image denoiser in action. And in this demonstration, I will be showing you how a mouse looks that is implanted with a chamber for in vivo imaging, and then how it is secured onto our intravital microscopy system. And then with the data acquired through this process, how the overall denoising process is done using the AI image denoiser. So here you can see that the mouse is secured and then it is mounted onto the intravital microscopy system for in vivo imaging. And once you've acquired data, you can bring it into the program. And when you click the execute button, the denoising process begins. And you can see how the process is being done through the progress bar at the bottom. And once the denoising process has finished, you can see the finalized denoise files on the left and also on the right top corner of the program. And you can also choose what kind of file type you would like to export your finalized denoise file. And at the bottom, using the dials, you can also edit the brightness and the contrast, as well as the sharpness of the denoise file for your research needs. And after you have done your further editing to match your wants and needs for your research, you click the export button to save and then export the edited denoise files into your desired folder. And at the bottom here in the two screens, you can see the before and after results of the denoise process. So the left is before and the right is a denoised data file after. And if you look closely here, again, on the left side, you can see the raw data file before denoising, and on the right, the finalized denoise file using the AI image denoiser. And here, using additional quantitative tools, we counted the cells in each data set. And before I go more specifically into this comparison, I'd like to give a brief explanation of what kind of experiment that we wanted to conduct with this data. So in the kidney, we wanted to visualize cell biodistribution and the localization of the cells, and also small microcirculation events that goes on inside the blood vessels of the kidney. So in order to do this, we used a transgenic mouse to label the cells in the kidney. And this mouse has the potential to express green fluorescent proteins in the cell nuclei. So it helped us to visualize the cells. And then using CD31 antibodies that were conjugated with fluorescent dyes, we injected them intravenously into the mouse to visualize the blood vessels. And also, in order to see the blood flow in the kidney, we additionally injected another type of fluorescent dye to visualize that blood flow inside the kidney. And so here on the left side, you can see the raw data before denoise, and on the right side, you can see the denoised data. And if we take a much closer look, you can see that on the left side, due to a lot of background noise, you can see that not only were the cells counted, but also background noise were counted because they were mistaken for the cells in the kidney. Whereas on the right side here, after eliminating unnecessary background noise, you can see that it was much more easier to accurately count only the cells expressed in the kidney on the right side. And furthermore, this is a quantitative analysis graph of the cell count data I have previously showed you. The left side is the cell count analysis graph of the raw data before denoising, and the right side is the cell count analysis graph of the denoised data. And on the left side graph, you can see that approximately 788 cells were counted, whereas on the right, there was approximately an average of 285 cells that were counted, which is a very big difference in numbers. And this again is due to the fact that on the left side graph, which is from the raw data, as I showed you before, not only were the cells counted, but also a lot of background noise was mistakenly counted as the cells. This leads to unreliable and inaccurate quantitative data. Whereas on the right side, after eliminating those noises, we were more specifically able to count only the cells, creating much more reliable data. And this goes back to the presentations that were done previously, taking into consideration the fact that noise consistently disappears and appears, whereas true fluorescent signals from the cells have very consistent patterns throughout the data set. So we were able to effectively distinguish the noise and the true signals of the cells using the AI image denoiser. And after eliminating those noises, we were able to only extract the true fluorescent signals of the cells in the kidney. And so by implementing the AI image denoiser before quantitative analysis especially, you are able to create much more reliable, replicable, and accurate data for your quantitative analysis research. So this is all for the brief virtual demonstration of the AI image denoiser. Currently, the AI image denoiser is available with a trial version. If you're interested in the trial version, please contact us through the email that you can see on the screen here, or also the link that Kubra has shared with us today. And thank you for sitting with us throughout this long session. And thank you again for joining.
Thank you, Jenny, for showcasing the IVIM's AI image denoiser software. And again, a huge thank you to Dr. Yoon and Dr. Kim for their insightful presentations. We now open the floor to questions from the audience. We already got two, and I'm gonna ask to Dr. Kim. In case of nanoparticles or exosomes, can we quantify them at one particular site at a time along with video capture? Also, can we capture multiple sites and collate them to quantify the biodistribution?
Well, okay, so let me answer it. So in case of nanoparticle and exosome, we can quantify them at one particular site along with the video captures. And then, so at one time, we can, at single time point, we can see only one single particular site. So if we want to see the multiple sites, what we need to do, we move around to, different positions with some time interval, and then see the biodistribution. So actually, this function is implemented in our microscope, which we call multi-position time-lapse video and image capture functions. So basically, we move around at different position with time interval and capture their videos. And by doing that, we can capture multiple sites at continuously and then analyze later.
Okay. And there is another question from the same participant. Do you think this technique can be used for orthotopic cancer models? You have shown the application for subcutaneous tumor models. Has it been tested before?
Oh, yeah, sure. So not just the subcutaneous tumor model. The pancreas tumor model I showed you was actually orthotopic pancreas tumor model. And then we also have a imaged orthotopic brain tumor model and orthotopic colorectal cancer model as well. So it's all possible. All different type of orthotopic tumor model can be imaged by using intravital microscope.
Mm-hmm. And we have another question about the support. Which data was the support model trained?
Okay, so for support model training, so we don't need a pre-training, but to accelerate the inference, we train our AI denoising model by using our real-time process videos obtained from all different kinds of organs, internal organs in a live mouse model. So as I showed you, we have been able to images almost every organ and tissue in a live mouse model with the process labeling. And then all those, video real-time, high, fast kinetic imaging data was used to train our support model.
Thank you. And another question is, what's the effective pixel size in the dynamic model? What is the physical resolution of the imaging?
Okay, so effective pixel size can be changed depending on the magnification of the objective lens we use. But to give you an idea, by using the high magnification objective lens, such as 25x or 40x, the pixel size will be will be 0.4 or 0.5, around 0.4 or 0.5 micrometer. Or by using higher magnification objective lens such as 63x, the pixel size will be 0.2 something micrometers. So that will be the effective pixel sizes. And then we know and then it depending on the number of pixels and the magnification objective lens, the effective pixel size it will changes, and the actual resolution will also changes. But normally it's in the range of the 0.5 micrometer is the typical physical imaging resolutions.
Right. And is this possible with skeletal tissues and organs, such as tendons and bones?
Yes, it's possible. Yeah, I didn't show you today, but we have imaged the tendons, exactly that Achilles tendons, and then also bones. In case of bones, we have imaged mostly cranial bones because that's easily accessible. And then by using two-photon microscope, we can directly see the bone structure based on the second generation signals.
Another question is, you have shown the 24 hours video. Maybe I didn't get it right, but has this video also been recorded from a living mouse? If yes, how is such a long anesthesia possible?
Yes, actually you are right. I obtained this 24 hours video from the living live mouse, and then for that we used the gas anesthetization, basically ice spray as our anesthesia.
And
then at the same time for 24-hour imaging, not just so for 24-hour live mouse images, we use ice spray for anesthesia, and then we also supply the water and nutrients through the catheter to avoid the dehydrations.
We have another question from Dr. Sam Pang, but we will need the rest of the okay, it's all right. I have noisy images captured by another brand's confocal microscopy. Is this AI software used for these images? How should we work on it locally? How much resource should take my computer for running your software?
Okay, well, it should be possible, but you need to export. You need to export this like a confocal microscopy video to the common video format such as MP4 or AVI. Then our AI software can deal with those kind of standard format videos. And then for computing power, you don't really need a high computing power. So normal desktop, normal common mundane desktop can do this processing.
And is the motion compensation software available as a standalone package?
We have a plan, but not now, not now. But in the near future, we have a plan to provide our motion compensation software as a standalone packages. Thank you for your interest.
Thank you for all the answers. At the moment, we answered all the real-time questions. And if you have any more questions, please send us through information@ivimtech.com, and we will be happy to answer them through email. And with that, we have covered all the questions. Thank you for your active participation. As mentioned at the beginning, we have just released the AI image denoiser software as a standalone solution. So you can now use it with other imaging data you already have. For any inquiries, as I said, feel free to contact us at information@ivimtech.com. Thank you, and we are looking forward to the next time.